Morning. Yeah, you can see I'm going for a like a light side, dark side. Split down the middle, kind of thing. I think if I rotate just right, yeah, there we go. The shadow kind of Phantom of the Opera's me, so that's pretty neat. Raw. Anyway, yeah. The first question is always fantastic. It's from Polly and Ollie again, who, yes, I continue to imagine as two little tiny blue parakeets sitting on a branch. Yeah, even though they are almost certainly not. But they're really good at typing for parakeets. I don't know. It's hard to say. Anyway, so it's Polly and Ollie here again. Fantastic. Yeah. Something that's been worrying me for a couple of days. So the gist of it is that their post lady is lovely, important, yeah, but also a heavy smoker. And so the post lady sets down a package on the front step and then backs off to 2 meters. And then they go out, pick up the package. But what she's saying is, I can still smell smoke because the post lady is a very heavy smoker. And even around the package, even though she's a couple meters away and she's not actively smoking, this is just left over from her breath, yeah, I can smell smoke there. Does that mean I could catch the virus if she had the virus? This is respiratory droplets. Now, what you're smelling is an aromatic. So it's a compound that's going to come out in the same way as the respiratory droplets. And it may be in those droplets. Actually, it probably is in those droplets. And so as the droplets, which are mostly water, are going to evaporate, all the things that can evaporate with them, like these little aromatic compounds, which are basically things we can smell, are going to evaporate too. And that's what you're smelling. And so, yeah, this is the idea between respiratory droplets. Now, that's the bad news side. Yeah, the not so bad news side is that it looks like via the intranasal route, at least in mice. So what I mean is some version of squirting or puffing virus up the nose of an animal. Looks like the infectious dose is somewhere in the neighborhood of 10 to the fourth or 10 to the fifth, which means that it takes somewhere between 10,000 and 100,000 individual virus hits to score a hit that actually starts an infection. Why is this, you say? Because you can go in other routes. If you're doing an animal experiment, you can inject the virus right into them, and yeah, it's a much lower dose needed to start the infection, maybe 10 times less than that. And you can go in, actually probably less than that, you can go right into the brain. For some experiments where they're trying to figure out what's the effect of a virus on the brain, you can inject it right into the brain, and in some cases, the virus is actually, it looks like it's either one virus or maybe a little less than one virus that starts the infection. And so you'd say, well, how can less than one virus start the infection? What are you smoking? Yeah. And the answer is that viruses are like this little cloud of misfit toys, basically. They make a lot of mistakes, and most viruses are not very good. Most individual virions, that's the little ball with the spikes coming out of it, are just not going to make it. They're not suited to this world or any world. And they have some kind of glaring flaw or defect that just means they aren't going to work. Viruses put themselves together, and I don't know, if you look at things that children put together, they're usually not very good. Yeah, no offense, my children. Like when they're young, I mean. Now they're actually better than the stuff I do, but whatever. Yeah. But imagine a virus that has no brain and is like 8 hours old trying to put itself together. Yeah, conceptually, that's about how it is. And so the word on the street and through some scientific tests is there's somewhere between 100 and 1,000. That's the normal range. Viruses that don't work for every one that does work. And so when I say that you can put it into the brain with a certain type of mouse coronavirus, and you get less than one virus actually starting an infection, it just means that some of those viruses that didn't work normally will actually work when you put them in. And the only way we can count these, well, there's a couple ways you can count the virus. You can count the individual particles on an electron microscope slide, but of course, by counting them, you've stained them with uranium and you've destroyed them with electron radiation. And so you can look at them, but you'll never know if they infect. Or you can try to infect something, but you'll never know how many there were, to start. Or you can try to split your sample in half and do it on both of them. That takes a little more time, and you can kind of estimate and say, okay, I looked at 3 samples and they all had this much virus. I tried to infect 3 sets of cells with the same amount of virus, and it caused this many infections. So, and you can also count the number of copies of the virus. Most coronavirus copies will only have one genome. It's a big long piece of RNA, all curled up inside like a big long spaghetti. And yeah, so you can count the number of those. You can count the number of those that kind of land on and stick to a cell, or you can try and scrub them off the outside. There's lots of ways to do it, but they're all a little bit indirect and somewhat unsatisfying, I have to say, as a virologist. But yeah, that's the reality of viruses, is that it takes 10,000 to 100,000 active viruses. So multiply that by, I don't know, 100 to 1,000. That's how much you actually have to breathe. So yeah, we're real good at picking up smoke. I don't know of anybody or even any dogs that are real good at smelling coronavirus yet, which I guess is good, because you'd probably catch it if you could smell it. And yeah, it's probably a little negative selection loop right there. Yeah, you could. There you go, boy. What do you smell? And then cough, cough. Yeah, in theory. Yeah, all this is in theory, because dogs do not seem to get severe coronavirus infections. Although there are a couple cases now where it looks like a dog might have been just passingly infected, but with no disease. So, yeah. Yeah, don't worry about your dog giving you the virus. Worry about you giving your dog the virus, or your cat, and keep yourself safe, and that keeps everybody safe. So, yeah, long way around. My gosh, that was a walk, wasn't it? Yeah, if your lovely post lady was infected, and you can smell the smoke there, that's a nice little marker. It kind of tells you that there are respiratory droplets there. Normally, you would need some kind of laser to detect respiratory droplets, and most people don't have the laser or the detection set up to be able to do that. And so, yeah, if you can smell somebody, you're in their cloud, you're in their thing. But that doesn't mean you're not with respiratory droplets when you can't smell it. They're also there if you're anywhere near a person. It's just a really nice illustration of where they are. So, yeah, you're probably okay picking up your post and saying hi to your post lady from 2 meters away. I feel like that is just fine. But yeah, there's a certain amount of risk that we live with every day. And not just COVID, but all kinds of viruses, and yeah, all kinds of things. And for the most part, we have mucus, and the mucus just hoovers these little viruses up and takes care of them. And thank goodness for snot. Maybe that is our big message for today. Yeah, thank goodness for snot. And thank you very much. This has been Ask Dr. Ben.